Hello and a warm welcome once more to Nesta's Bioacademy. And today we are going to go through our third lesson on osmosis, with the main objective of this lesson today to demonstrate osmosis. So our main objectives today are to, one, demonstrate osmosis and also explain the various experiments which we always use to demonstrate osmosis. We are going to look at the various ways we can always carry out investigations in our laboratories to check whether osmosis has taken place or not. And again, remember, this is a highly examinable area in biology practicals, so it's very important that you understand the various ways in which these experiments are always examined. And the requisite knowledge you need to know by now, still is need for you to have a firm grip of what osmosis is, the direction of movement of water, that is from a dilute solution to a concentrated solution, the tonicity of the solutions, hypertonic, hypotonic, isotonic. So there's need for you to be able to have a firm grasp of that. Remember in the previous video, we learned about what happens to plant cells and red blood cells when they're placed in solutions of different concentrations. And we also learned the important roles osmosis plays in living things. So in case you missed that, you can get to check on that before you proceed with this video. Remember, one critical examiner tip I need to highlight for you is when we ask you to describe the role of osmosis in living things, you must pay attention. It can be state 3 roles of osmosis in living things, state 3 roles of osmosis in plants, or state 3 roles of osmosis in animals. So you always have to pay attention to the stem of the question so that your response is well engineered to meet the demands of the examiner. Now, osmosis can always be demonstrated using a number of artificial semi-permeable membranes. For example, you can always use the Visking tubing, the cellophane paper, or the dialysis tubing. We can also use soft plant tissues like the soft stems. We can use the fruit peels to also investigate osmosis. And also you can use plant organs like the raw potatoes, the raw Irish potato cylinders can also be used. And of course, you can also use eggshells to illustrate osmosis. So in this lesson, we are going to look at the various ways through which this can be achieved. So experiment 1, we are going to review the Visking tubing experiment. This experiment usually requires the following materials. Visking tubing. You can be given 2 solutions of different concentrations, and in a practical exam, you might not know which one is which one is hypertonic, which one is hypotonic. You can be provided with the threads, and you can also be provided with a glass rod. And once you have these materials, you might be instructed to set up your apparatus as shown. Okay, this is illustrated. You already know what you are having here, but sometimes in exams, when you're given 2 solutions of unknown concentrations, from the instructions, you'll be told to place one in the beaker and one inside the Visking tubing. The Visking tubing is always flattened, and for you to use it, it's always recommended using the piece provided to you. You dip one end, you can dip it in water, and then once you remove it, you rub it between your fingers, and that will always open up the Visking tubing. So once you have set up your apparatus as shown below, you can always be told to allow it to stand for a few minutes. And what are the kind of questions, most common questions you can meet from this experiment? The examiner can demand that you state the observations that will be made. It might be required that you explain the observations that you've made, and you can also be told to identify the physiological process under investigation and probably highlight the various roles that that physiological process you have identified plays in living things. So I remember from our earlier video when you're asked to account for the observations made, there's a particular way of responding that you always need to have of knowledge of to always increase your chances of scoring well. So like the observation that would be made here would be that the Visking tubing contents increase in size. Okay, like here we can see we are having distilled water in the beaker and we're having 20% sucrose solution inside the Visking tubing. Be open-minded because sometimes it might be the reverse. You might be having distilled water in the Visking tubing and the 20% or any other concentration of sucrose solution or any other solution inside the beaker. So in this case, we are expecting the observation to be the reverse. We expect the Visking tubing contents to decrease in size, or we expect the Visking tubing to decrease in volume. So the way the question is framed, you have to compare. You have to look at the two solutions on either sides of the Visking tubing, and you have to understand which one is hypotonic to the other, and then that way you will be able to tell the direction of osmosis, and then from there you'll be able to tell whether there will be an increase in volume or a decrease in volume. And remember, from the way the setup is placed here, the main observation will be the change in volume of the Visking tubing, not on the level of the distilled water in the beaker. Why would try to think of the reason why you are not, like in this case, try to think of the reason why you are most likely not going to see a change in the level of the distilled water in the beaker. When you are to account for the observations made for the 3 marks, one, you need to compare the concentrations of the two liquids given. So distilled water is hypotonic to the 20% sucrose solution. So water molecules will move into the 20% sucrose solution by osmosis, second mark, leading to increase in contents or the volumes of the Visking tubing for the 3rd mark. So you can see clearly there's a comparison here. There's mention of the process taking place, and it is all related back to the observation made there. Comparing, as we said earlier, it can be done the other way around. One can say the 20% sucrose solution is hypertonic to distilled water. So the water molecules move into 20% sucrose solution by osmosis and leading to increase in the content or volume of the Visking tubing. And that is how those marks are earned there. That is the response that should meet the examiner's demands in an effective way. Another very common experiment you're likely to find here is where we use the raw potato experiment. So here we will give you a raw peeled potato, and then the setup is set up as shown here. We can have distilled water in the beaker, and then we're having the raw peeled potato. Then you can have concentrated sugar solution in the potato. There are sometimes when instead of the concentrated sugar solution, you might be, we might have a case where we place a few sugar crystals inside the peeled potato. So remember, the potato is raw, it is peeled, and then we create some depression in it to hold onto to hold the concentrated sugar solution, or to hold the sugar crystals that are placed there. And what will be the most common questions here? Again, you can be asked to state the observation made, and then you can be asked to explain the observation that has been made. And mostly these would be tied questions, meaning if you state a very wrong observation, then the explanation will obviously be wrong. Just like in this earlier, in this other case that we looked at, if you mess with the observation, then obviously accounting for the observation also becomes very tricky for you to score, because the questions are always tied. State the observation made, 1 mark. Then part 2, account for the observation you have made above. Meaning if you state a wrong observation, it becomes difficult for you to score for the explanation as well. So coming back here, we can also ask you to explain what will happen if we were to use a boiled potato instead of the raw one. That's a very common question also you're likely to meet here. So what would be the observation made here? The observation made here would be that the level of concentrated sugar solution will rise, or there will be an increase in the amount of the concentrated sugar solution, 1 mark. Then if we are to account for the observation made here, then the explanation would be that, 1, the explanation of that distilled water is hypotonic to the adjacent potato cell subs. So water molecules move into adjacent potato cell subs by osmosis. Consequently, the adjacent potato cell subs become hypotonic or dilute compared to the next cortex cell subs, next potato cell subs, not cortex, but potato cell subs. So water moves into the next potato cell subs by osmosis, and then this continues until water reaches the concentrated sugar solution, leading to rise in the level of the sugar solution. One thing is evident here. Remember, when we have a potato like this, here we are having hundreds, thousands, and possibly even millions of cells. For if distilled water were to move from here all the way to the concentrated sugar solution, it will be passing through numerous and numerous potato cells. And therefore, it's always ideal that when responding to this question, you indicate the cell-to-cell movement of water. We are not going to treat this as a single semi-permeable membrane, like in the case of the Visking tubing that we are using, we have observed in the previous experiment. So a key examiner tip here is always that you should not always treat the entire potato as a single Visking tubing or cell equivalent. The cell-to-cell movement of water needs to be well described. So that is the explanation to always note there. Distilled water will always be hypotonic to the adjacent potato cell subs. So because of that one, we expect the water to move to the adjacent potato cell sub by osmosis. But then remember, we have now the sequence of cells heading all the way to this end. So once the water molecules move into the adjacent potato cell subs, this will therefore mean the potato cell sub here now would be hypotonic or dilute compared to the next potato cell sub. So water moves from the adjacent, from the first potato cell sub to the next potato cell sub by osmosis again. Then you will find that this process continues until water reaches the concentrated sugar solution, increasing the level there. So that is a common question you're also likely to meet And do not just say that distilled water is hypotonic to the adjacent potato cells up. So water moves to the concentrated. Don't just say that distilled water is hypotonic to the concentrated sugar solution. So water molecules will move to the concentrated sugar solution by osmosis, leading to the rising level. An explanation like that one will not bring in the cell-to-cell movement of water that the examiner might demand that your explanation brings out effectively. Then we have this other case where we use soft tissues or plants like a stem. And a very common case you can have is we can give you softer stems of plants. Most of the times we can even give you stems of Tradescantia, like the one you're seeing here. You can also give you the petals of kale, that is Sukuma wiki, and of course, many other soft stems of plants can be used. The ones which are still young and relatively flexible can be used. The very rigid stems will not bring out the observation well. And if I can explain this briefly, here, even if we were to give you the Tradescantia, you might be expected to cut off maybe 2 centimeters from this end, and then when you cut it out, you will obviously get something of this nature. The outermost layer of this piece of stem will be having the epidermis, and the epidermis is always covered by cuticle. One thing to note about the cuticle is always that it is always impermeable. It is always impermeable to water, and this is important because it enables the plant to minimize water loss through transpiration and evaporation from the plant tissues. Then on the inner side, we have a tissue we always refer to as cortex, made up of many cells. So again, if I to bring this here, sometimes actually exams will always mind mostly that you cut the stem into 2 halves, longitudinally like this. So on the innermost side, so from the cutting, it will be something of this nature. If I can represent it in a 3D way, with this being the line of cut. So when you cut it into 2, the innermost end will have the cortex cells. Then on this end, we have, of course, the epidermal cells there. And you can see they're covered by a cuticle, which is always relatively impermeable to water. So this is going to have some profound effect on the observations we are going to make. And that's why it's important for you to understand this, that the outermost end usually has a covering of cuticle, which is always relatively impermeable to water, but the innermost cortex cells are not covered by the impermeable cuticle. This essentially means that the innermost cells, the innermost cortex cells, like in this case, they will easily gain more water. So these ones will easily gain more water, but these ones will gain less water. In terms of losing water, also they will always, these ones will lose more water, and these ones are likely to lose water at a lesser rate. So again, you can be asked to make a stem cutting, cut it into 2 halves longitudinally, and then you place the 2 cut halves into different solutions. One solution will be probably hypertonic, the other one will be probably hypotonic, and possibly another one can be isotonic, or you can be told to leave one as a control, not placed in any of the solutions. Again, the most common questions here that you'll be asked will be to state the observations made after some time, to account for the observations made in the 2 different solutions, to name the physiological process under investigation and also to describe the role of the physiological process under investigation, either in plants, in animals, or in living things. So let's assume we were told to place these two cut halves in two solutions, solution L1 and solution L2. And then from the drawing, this is the epidermis and the inner side is the cortex. Then if you're to make your observations after like 20 minutes, these are the observations you see. How will you describe your observation? What will you say has happened here for L1? And what will you say has happened for L2? Ponder over that, pause over that one, and ponder over it. What can you say has happened? Initially, you placed straight portions of the cut stems, but now look at the observations you are seeing. So how will you describe these observations? Okay, so moving on, let's look now describe them together. The piece in L1 is said to curve outwards or in the direction of the epidermis, but the piece in L2 is said to curve inwards. Okay, it makes sense this way. If, remember, if I can do this illustration here, remember this one is originally the stem, piece of stem. Then we have cut it into halves along this length here. And then later on, so this is the epidermis. And this is the cortex. Now, if we look at this is the epidermis. The epidermis is the outermost end, and the cortex is the inner. So the epidermis is always the tissue that is protecting the internal surfaces. So basically, epidermis is the outermost layer for the stem. The cortex is found to the inside. So curvature can happen in two ways. You can have a case whereby curvature imefanyika hivi, with this being the epidermis and this being the cortex. Alternatively, we can have a case where curvature happens this way, with this one now being the epidermis and this being the cortex. So in this case, you can see the bending is happening in this direction towards the cortex, and the cortex was on the inner end. That is why we are saying this one is curving inwards. And here you can see the curving is happening towards the epidermis, which is to the outermost end. That's why we are saying it is curving, it curves outwards. So you'll always find us referring to those observations largely here. So the piece in solution L1 is said to curve outwards, and the piece in L2 is said to curve inwards. So remember, if the procedure was not detailing that, instructing you to feel the pieces, it will be wrong for you to give the observations in terms of one piece feeling rigid or one piece feeling less rigid. And again, if you are not told to measure the length after some time, you do not bring in observations in terms of length. So in this case, curvature would be the best observation to see in this scenario. Then you can also ask you to account for the observations made here. For these observations to be made, for piece in L1 to curve outward, it will mean that solution L1 was hypotonic to the cell subs, correct? So the cells gained water by osmosis, but the inner cells gain more water than the outer cells. So the inner cells will stretch more than the outer cells, hence the curvature outwards. Remember, we said, though we have Beyond the cuticle, we are having cells on this region, but because they are covered by a waterproof cuticle on this end, the rate at which these cells are absorbing water would be less compared to the rate at which these inner cortex cells will be absorbing water because they are not covered by any waterproof cuticle. So that is the explanation to give there. The solution L1 is hypotonic to the cell subs. So the cells gain water by osmosis, but the inner cells gain more water than outer cells because the outer cells are covered by cuticle, and the inner cells therefore will stretch more than the outer cells, hence the curvature outwards. Coming over to L2, solution L2 is hypertonic to the cell subs. So the cells will lose water by osmosis, but the inner cells will lose more water than the outer cells, and this is going to lead to the curvature inwards because the outer cells, remember, they're covered by a waterproof cuticle. So the rate at which they're going to be losing water is less compared to the inner cells here. So that is how you explain the observations for this case. And again, the examiner tip here, what is always most important for you to understand again here, when you talk of losing water, the tissues or the cells lose water. Don't say the cells loses water. This one is incorrect. And again, avoid referring to the fact that the cells gained the solution by osmosis, because when you talk about gaining solution, that is now not osmosis, because osmosis is exclusively movement of water molecules or solvent molecules, not solution. Again, critical for you to understand that. Another possible experiment, now over to experiment 4, a possible way in which this osmosis can also be investigated is by use of fruit peels. Here we can, the examiner can give you like a raw banana, and then using a scalpel, you are supposed to cut out portions of the peel. One precaution always with this experiment is that you need to always identify the part of the peel that is appearing to be straight. If you are to cut out a piece of the peel that is already having some curvature, then already your observations are going to be compromised. Another precaution always is to always use thin peels. If you use very thick peels, it might take too long, or sometimes you might not observe the curvature in any given way. The explanation is exactly the same as that of the previous experiment we have just investigated, only that now for the case of the fruit peels, the outermost layer of the fruit peel is always referred to as either the epicap or the exocap. And the inner cells here, this inner part is always referred to as the mesocap, also made up of cells. So again, in a similar way, the epicap or the exocap is always covered by a waterproof cuticle, while the mesocap is always, the cells are not covered by a waterproof cuticle. So it means, therefore means again, that the cells of mesocap will always gain more water, or they can lose water at a faster rate compared to the cells, the outermost cells, which are covered by the cuticle there. So the explanations always remain the same. And again, the examiner tip I've given earlier must always be adhered to. Don't say the solution loses water, losing or becoming less tight. And also don't refer to the cells gaining solution by osmosis. That would be inaccurate. Now, so again, still here, remember we're still referring to curving inwards or curving outwards. Like in this case now, this is the epicap. So this is, in this case, the curving is inwards if it is in the direction of the mesocarp, and this one curving is outwards if it is in the direction of the epicap there. Again, that's another very common experiment you're likely to encounter. Now, take note that other than the direct observations, the candidates may also be told to fill the specimens in either of the two solutions and then make observations. In this case, you're supposed to make observations based on firmness or rigidity. With a piece that has been placed in hypotonic solution, expected to always be very firm or very rigid, while the piece in the hypertonic solution is always expected to be less firm or less rigid after losing water, because the cells shall have become plasmolized. So they end up becoming less firm. So again, that is crucial to understand. Another possible experiment you're likely to encounter here is one in which we are using potato cylinders. So here you can be given a potato cork borer, and again, raw potato. It can be washed, and then after that one, using the potato borer, you will cut out and remove potato cylinders from it. And then you can cut out a given length of the potato cylinders, and then you place them in different solutions. After that, after some time, observations here can always be made in terms of change in length, it can be in terms of change in mass, or in terms of rigidity. Most common observations will always be in terms of change in length and also in terms of change in rigidity, like what you can see in this illustration here. If we cut out 3.0 centimeters of potato cylinders and place them in different solutions, you can observe that averagely, you can always find the pieces placed in solution A, giving you an average length of 2.7 centimeters. Maybe let's say in solution B, you get 3.0 centimeters, and the ones you placed in C being 3.2 centimeters. For reliability of these investigations, you are always required to place more than one cylinder. You can place like 3 cylinders in each case, and then later on you'll obtain the lengths and obtain an average for the same. If we look at for the first case, after some time, now the length is 2.7 centimeters. This infers a decrease. This means there has been a decrease in length. And for there to be a decrease in length, it means that the cells here lost water by osmosis. So under what conditions would the potato cells lose water to the solution? This will mean that the potato cell subs therefore must have been hypotonic or dilute compared to the solution. So your explanation is supposed to be that solution A is hypertonic to the potato cell subs, or you can say the potato cell subs are hypotonic to solution A. So the potato cells lost water by osmosis, then they shrunk and decreased in length. You shall have fully met the description for comparison, mentioning of the process that has occurred and the observation there. For this case, you can see the length has remained unchanged. This will mean there was no net osmosis, and therefore this would mean solution B is isotonic to the potato cell subs. So no net osmosis occurred. So the length of the potato strips remains the same. For C, you can see again, there has been an increase in length. This means there has to be intake of water by osmosis, making the cells to stretch slightly. So this will mean solution C is hypotonic to the potato cell subs. So the potato cells gained water by osmosis, and the cells stretched and increased in length. And that is how these experiments are supposed to be explained. The next table here gives a summary of these observations. and how they can be explained. That is, if length was the main parameter we are using to make observations, if we are using the level of firmness or rigidity, and if we were to use to have a means or a weighing balance to determine the masses, those are the explanations we will be expecting for both solution for solution A, solution B, and solution C as well. So the various mistakes to always be very wary of in these experiments are, number one, as we have pointed out earlier, it is going to be wrong to say the solution loses water or the cells loses water. The correct biology and correct statement and science there is loses water. Again, it is wrong to say the cells gained solution by osmosis. We have stressed on that already. And again, when you are comparing for the potato strips, it is wrong for you to say the solution is hypotonic or hypertonic to the potato strips or cylinders. As I said earlier, these terms are hypotonic, hypertonic, isotonic are terms we are using to compare concentrations of solutions. But is a potato strip a solution? Is a potato cylinder a solution? No, they are not. Instead, the cells making up these potato strips, inside these cells, we are having the cell subs, which contain fluids representing solutions there. That's why we always say the solution is hypotonic to the potato cell subs. The solution is hypertonic to the potato cell subs, but we don't say the solution is hypotonic or hypertonic to the potato. So again, that is important to understand. So again, further exploration, there is a need for you to always understand the basics of scientific experiments. Anytime you are investigating something, you will always be dealing with variables, so many variables. So they're always dependent variables. They're always independent variables. And then we always have the variables that you must always keep at a constant. So the questions, the examiner might seek to know what variables did you keep constant during the investigation, and it's always needed for you to be able to explain them. In addition, you can also be asked to identify and set up control experiments, which is important to know. Like in the experiments, you're dealing with potato strips, the raw potato we used earlier, the one in which we made a carving and placed concentrated sugar solution. A very common control experiment will be to use a boiled potato, because by boiling, you end up destroying the semi-permeability of the cell membranes, and therefore there will be no observable changes being seen, because water will not be able to move through the semi-permeable membranes to bring out observable changes. So experimental biology is very integral, is an integral aspect of biology examinations, and any student who wants to do so well in biology must always have a very firm grip of the experimental biology. And again, remember always that osmosis is movement of water molecules. It is not movement of solution. And that brings us to the end of this review again for the experiments there. If you have any question or something you wish to seek clarification on, kindly comment and I'll get back to you. Thank you so much, and let's meet in the next video where we'll be looking at active transport. Bye-bye.